India has once again made its mark in the global agriculture sector by achieving a major scientific breakthrough. The country has become the first in the world to officially develop and release genome-edited rice varieties. These varieties DRR Dhan 100 Kamala and Pusa DST Rice 1 were formally launched by Union Agriculture Minister Shivraj Singh Chouhan at a ceremony held at ICAR Subramaniam Auditorium, NAAS Complex, New Delhi. According to the Agriculture Minister, the introduction of these genome-edited rice varieties marks a major step forward in the modernization of Indian agriculture. These new rice types are especially designed to increase productivity, conserve water, and enhance climate resilience. Developed by scientists from the Indian Council of Agricultural Research, the varieties are seen as a significant move toward ensuring food security in a changing climate. The minister also felicitated the scientists involved and emphasized that these innovations reflect the vision of Prime Minister Narendra Modi's goal of a Viksit Bharat. He stated that Indian farmers are progressing towards prosperity through science and innovation. Now, what makes these rice varieties revolutionary is the use of CRISPR-Cas9 genome editing technology. Unlike traditional genetically modified organisms that involve inserting foreign DNA, genome editing involves precise changes to plants' own DNA without introducing any external genes. This method is considered faster, more accurate, and safer than older techniques such as random mutagenesis, which often involves the use of chemicals to create genetic changes. According to scientists, genome-edited crops like these can be developed quickly and are better suited to withstand environmental stresses such as drought and salinity. These new varieties promise higher output, reduced water usage, and lower cultivation cost, making them ideal for sustainable farming. While there was some concern raised in a recent report regarding the public acceptance of genome editing technologies, particularly the rDNA techniques, the Indian government has clarified its position. The Directorate of Knowledge Management in Agriculture has rejected these concerns, stating that two categories of genome-edited plants, SDN1 and SDN2, are exempt from strict biosafety regulation due to their proven safety. The DKMA director emphasized that over 30 agriculture-based countries have adopted similar exemptions and that these genome-edited crops are scientifically safe. There were also worries about whether these new rice varieties would affect Indian rice export, especially to the European Union. However, officials clarified that both DRR Dhan 100 and Pusa DST Rice 1 belong to the non-basmati category. The EU imports only an eligible amount of non-basmati rice from India. If concerns do arise, the Indian government is open to discussing the matter with EU stakeholders to protect the interests of Indian farmers and consumers. The development of these varieties is not just a national achievement but also a global contribution. With the growing challenges of climate change and food insecurity, India is working toward becoming a global food basket by increasing nutrition output, ensuring food security, and supporting both national and international demand.